Okay, we'll go ahead and get started. Welcome, everybody. It's a pleasure to introduce today's guest, Dr. Nazish Syed. Dr. Syed is an instructor at the Cardiovascular Institute at Stanford University. He started out doing his undergraduate medical education, getting his medical degree at University of Bombay in India, and then went on and did postgraduate medical training, including an internship and a residency, initially at the University of Mumbai and then at the Mahatma Gandhi Memorial Hospital. And from there, he came to the States to do a Ph.D. in pharmacology and physiology at the University of Medicine and Dentistry in New Jersey. And then Dr. Syed did a postdoctoral fellowship at Columbia in physiology and biophysics, and an additional postdoctoral training in cardiovascular medicine at Stanford, where he successfully competed for an independent NIH F32 individual postdoctoral award. And then he spent time in a place I used to hang out in Houston. He was working at the Houston Methodist Research Institute in cardiovascular science, and there he was recognized with the President's Award at Houston Methodist. He returned to Stanford and assumed his current position as an instructor in the Cardiovascular Institute. Dr. Syed's work has been recognized. He's been very successful getting grants early in his career. He received an American Heart Association Scientist Development Grant when he was at Houston Methodist, and then also took that to Stanford, I believe, right? And then he also won a Society for Vascular Medicine Kaufman Young Investigator Award. He currently has about in the middle of a 5-year NIH KO1 award that he got from the National Heart, Lung, and Blood Institute, and that particular award's on endothelial dysfunction and dilated cardiomyopathy. And he also serves as a co-PI with his lab chief on a couple NIH R1s at the moment. And he's been a very prolific publisher, has around 40 or 40 plus papers, and a lot of them in really top journals, including Cell, PNAS, Translational Medicine, Frontiers in Cell and Developmental Biology, Cardiovascular Research, et cetera. Dr. Syed's work early on, he actually worked on something very dear to my heart, nitric oxide and modulation of soluble guanylyl cyclase, and looking particularly at the post-translational modification of S-nitrosylation. And some of that work is very important and seminal, not only in cardiovascular science, but in neuroscience as well, and work that I had followed. And then he went on and was studying reprogramming somatic cells to iPSCs, and particularly looking at the role of the innate immune system in that transition, which has really, I think, moved that field in a very important direction. And most recently, he's been using iPSCs to model human vascular diseases, particularly laminin-associated vascular dysfunction. And he's looked not only at genetic disorders, but it induced vascular dysfunction that occurs, for example, after chemotherapy or after diabetes-induced vascular dysfunction as well. So we're going to hear a bit about some of that work today, and it's my pleasure to welcome you.
So, good afternoon, everyone. I want to thank Dr. Friedlander and Dr. Gordy for the invitation. It's a real pleasure visiting Virginia Tech. And thanks for the introduction. So I'll give a little bit about my background, what I worked on. Dr. Friedlander mentioned about my graduate work on nitric oxide. And then I'll try to show a little bit about my postdoctoral work and with the overall scheme of vascular dysfunction and what we have tried to do for endothelial regeneration, and then talk about the use of stem cells in different diseases. So this is a little bit about my background. I did my MD back in India, followed it by my residency in Mumbai. And as Dr. Friedlander mentioned, I did my PhD from Rutgers, New Jersey Medical School, where we basically try to look at the role of soluble guanylyl cyclase, which is the nitric oxide receptor, and its role in nitrate tolerance. I was fortunate that I was able to publish some fundamental work on its role in nitrate tolerance, a clinical feature which we don't know much about it, and how a post-translational modification can result in vascular tolerance and nitrate tolerance. I would not go in detail about this thing, though I would love to when I move forward from nitric oxide, but I still think somehow or the other we can try to connect it together because it's been produced by the endothelial cells, having an effect on smooth muscle cells. But I'll go briefly on my introduction. I followed this with a postdoctoral fellowship at Stanford, where we were trying to understand how stem cells can be generated from different cell types, from fibroblasts, from blood cells, but how we can make this an efficient reprogramming. So if you reprogram fibroblasts, which are skin cells, the efficiency is very low. It's about 0.001%, which makes it that it's a very low yield. My initial work as a postdoc was to understand somehow or the other we can improve the safety, but also its efficiency of making iPSCs. This was about 8 years ago, so we have made great progress around this thing. But one of the main aspects which we figured out is that if you do efficient reprogramming using viral vectors, and I was having a conversation with Jamie that how viruses have a much more efficiency of reprogramming or infecting because it can hijack the transcriptional machinery of the cell, then why do using cell-permeant peptides have a low efficiency of reprogramming? So what we did in this summary of 3 years of work in this one slide is that is there any way we can try to learn from the viral system of how it brings about reprogramming and then translate that work in using cell-permeant peptides. So I'll briefly summarize this, that retroviral vectors or the lentiviral vectors which are used for reprogramming uses the innate immune system for hijacking the system and then utilizing it for spreading it. We did exactly the same thing. We used the toll-like receptor 3 model system where we use cell-permeant peptides which can stimulate the toll-like receptor 3 and then improve efficient reprogramming by bringing about chromatin remodeling. Because innate immune system is about bringing about chromatin changes with this thing. As I said, I'm summarizing it in very briefly. It's not justice to this paper. We published this in 2012, and then we followed it up that if we can make these in iPSCs, is there any way we can transdifferentiate one cell type to the other cell type without the use of an intermediary induced pluripotent stem cells? And this we could do it by taking these stem cells, modulating the cell type by stimulating with innate immunity, having a chromatin which is a much more open chromatin, and then provide the growth factors which we can push from one cell type to the other cell type. I mean, my primary cell type was endothelial cells. And if you can think about, we can directly convert skin cells, which are fibroblasts, to endothelial cells. This could be a great model system for myocardial infarction, where most of these cardiomyocytes are turning into fibroblasts. Is there any way we can convert these fibroblasts, which are scar cells, into endothelial cells? It will help in the recovery of in myocardial infarction. So that was part of my postdoctoral training. We published this paper. This is a brief paper just showing how we can convert fibroblasts to endothelial cells without having transcription factors, but just stimulation of the innate immune system. Briefly, just showing some of the animal work. But what I want to really focus on today about my talk is that how these stem cells or induced pluripotent stem cells can be used for disease modeling, can be used for regenerative medicine, and importantly, how it could be used as a tool for drug screening in a dish, which could then be applied for clinical trials, where we are not going to expose certain drugs to patients. We try it in a dish, see if they are improving the cell type, improving the outcome of the cells, then before giving it to the patients. That's what my talk is going to be. I'm going to try to give this brief summary of how pluripotent stem cells can be used for drug screening, can be used for disease modeling, and if both of these are successful, can we use it in a 2D or a 3D model, so kind of getting a little closer to the human, and then apply it into the patients. I'll try to cover all aspects of it. As I said, I'm a vascular biologist, so I'm a little more biased towards endothelial cells and vascular cells, and I think they contribute much more to the heart than the heart cells itself. And that's why I put this slide up, that all roads lead to Rome, because if, and I always give this analogy, if your kitchen sink is leaking, your first thing you should look at is your plumbing, which is your vasculature. And if you can fix the vasculature, there's a good chance your kitchen sink is not going to leak. So, importance as a cardiologist, as a clinical cardiologist, I always thought it's the cardiomyocytes which need to be looked at. But my postdoctoral training in vascular biology taught me one thing, is that the vasculature plays an equally important role in the heart disease as much as the cardiomyocytes. So based on that, what I'm going to talk today about, that was a little bit about my background, is how we can model endothelial dysfunction in dilated cardiomyopathy, a disease of the heart, by actually not modeling the heart itself, but modeling the vasculature, which is 3 times more than the heart cells. And what different tools we can apply to actually have a small clinical trial in a dish, which could be implemented in 2 of my patients, which we were able to give the drug. So a little background about dilated cardiomyopathy. I mean, you guys probably know about it. It is a disease which leads to a reduced capacity of the heart to pump blood. It is caused by a number of mutations. If you look at right here, there are 60 different mutations, but I wanted to focus on one disease, one specific mutation, which is LMNA or lamin A. Now, the reason I focused on this is because it contributes 6% of all the mutations which lead to dilated cardiomyopathy. So making it obvious that there are more patients suffering from a lamin mutation, which leads to dilated cardiomyopathy. A little bit about lamin. So it is referred as 1 gene, 2 protein, and multiple diseases, because mutation in 1 gene can lead to different kinds of diseases. Lamin is a very key gene, and if you look at this, between the cytoplasm and the nucleus, any information which needs to pass through the nucleopore complex has to go through these strands, which is the lamin AC. And any information leading to chromatin changes has to pass through this lamin A and C gene, which then conveys message to the nucleus saying that a chromatin change is required, epigenetic change is required for the cell to behave differently. And as I said, it's 1 gene, 2 proteins, but multiple disease. And if you have a mutation in a lamin AC gene, you could see 11 different kinds of diseases could be associated with this mutation, including muscular dystrophy, including progeria, which is an aging disease where these individuals age, even if they are 15 years of age, they look like about 80 or 90 years of age. It is because of a lamin AC mutation. So it became a easier target for us to study. But the most important thing is that these patients were easy to get. We had in Stanford Clinic, there's a majority of these laminopathy patients which were coming in. So as I said, laminopathies is at the center of the heart disease. Mostly all of these 10 different diseases have some kind of a cardiac link. Unfortunately, most of these patients die because of a cardiac involvement, even if it's muscular dystrophy or lipodystrophy. Their ultimate demise is because of a heart disease. And if there any way we can try to prevent that, we might be able to extend their life and quality of life, basically. But as I said, we are quite myopic when we think about us as cardiologists. We think about the cardiomyocytes, but I wanted to look beyond the heart in this study and start focusing on the other 2 cell types, such as the cardiac fibroblasts and the endothelial cells. And if sort of we can improve the endothelial cells or the vascular cells, is there any way we can improve the cardiac contractility, which is impaired in dilated cardiomyopathy? Remember, the dilated cardiomyopathy, the heart is beating at a much slower pace. It doesn't have the capacity to pump blood outside to reach to the different areas. So this would be my scheme of the presentation. I will start from the patient itself, and in this 40 minutes, I'll try to convince you guys that studying this patient by using iPSCs and iPSC-derived cardiovascular cells, we can model the disease. We can understand the different aspects of the mechanisms. If we get lucky, we can screen for drugs if we can identify a target, and then customize the medical plan and give it back to the patient to see if it can improve the health of this individual. And that's what we have tried to do as a clinical trial in a dish kind of thing. And to answer these, we had 3 specific questions for our hypothesis. One is to characterize iPSC-derived endothelial cells from these DCM patients. Second is, if we are able to characterize them, can we figure out the mechanism? And the third simple question is, if we do know the target and the mechanism, can we screen for some drugs in the dish which could be implemented in the patients? And to do so, as I said, one of the most easiest things for us in Stanford is that we had a whole 4-generation family suffering from this mutation. all live in the Bay Area, and this gave us easy access to get hold of these patients, get blood samples. For some of them, we were lucky to get heart samples as well. So as I said, this is a 4-generational family having the mutation. All the red dotted ones are the one which has the mutations. They have a clinical phenotype. They all come to Stanford Clinic. All of them have dilated cardiomyopathy. We selected 2 of the non-mutation patients as our internal control, as healthy control, because they do share some portion of the genome. The mutation was a frame shift mutation, so it led to haploinsufficiency. There is reduced production of LMNA protein, which lead to preventricular contractions and eventually leading to dilated cardiomyopathy. You could see the mutation is present in all of them. They all have a cardiac phenotype. But what we started to investigate is that in addition to a cardiac phenotype, they actually all had a vascular phenotype too. They all suffered from hypertension and in massive atherosclerosis. So that led us to think that, do these individuals have endothelial dysfunction in addition to their cardiac phenotype? So for that, what we did is we recruited these patients back in the clinic, and we measured their endothelial function clinically. You can measure this with a device called as EndoPad. It's like a echo machine for your vessels. It's a 20 minutes of non-invasive technique where you can occlude one of the arms and then compare it to the non-occluded arm and see whether there is an increase in flow-mediated vasodilation. The way your endothelial cells are always in contact with flow-mediated blood. The blood is always gushing through your arteries, and your endothelial cells are always in a state of dynamic changes. But if you occlude it for 5 minutes, there is static blood in the endothelial cells, and when you remove the flow or the occlusion, the gush of the blood flowing through the endothelial cells can be detected by the computer, which then calculates by an algorithm and spits out a value which can determine whether it's endothelial dysfunction or not. So it's based on reactive hyperemia index. It measures the amount of blood flowing through the lower arm. And it says if it is more than 1.67, then it's normal endothelial function. But anything lower than 1.67 is considered as abnormal endothelial function. A lot of hypertensive patients, for chronically hypertension patients, suffer from endothelial dysfunction because of turbulence in their blood flow. A lot of massive atherosclerotic patients have endothelial dysfunction. So we used this method and we said, let's try to see whether our cohort has endothelial dysfunction even before we go ahead and we differentiate their iPSCs into endothelial cells. And indeed, you could see right here, the 2 control patients had normal reactive hyperemia index, more than 1.67. 3 of the patients volunteered to come and do this test repeatedly, and you could see, and I've just labeled them as patient 1, 2, and 3. You could see they all had repeatedly a reactive hyperemia index of less than even 1. So they had massive clinical endothelial dysfunction. Just to make sure that this equipment is measuring correctly, we took a chronic hypertension patient, and you could see, as I mentioned, he also had endothelial dysfunction. So we knew that these patients, in addition to having a cardiac phenotype, have clinical endothelial dysfunction. This led us to think that now is the time we can try to take these patients, make iPSCs, which are stem cells, and now do a whole characterization in the dish. So to give you a little bit about the background about this, I'm sure you have all known about what induced pluripotent stem cells are. It was pioneered by Dr. Shinya Yamanaka, for which he got the Nobel Prize. It basically does is it turns back the clock. It can revert your skin cells to to stem cells. In our normal development, it's the embryonic, the blastocyst, which differentiates to different cell type. Here, by forced expression of 4 factors, which were called the Yamanaka factors, you can turn these skin cells, which are terminally differentiated cells, to stem cells. Now, these stem cells have all the characteristics of embryonic stem cells. However, they are adult stem cells. They can regenerate. They can differentiate to whatever cell type you want, and they are pluripotent, that they can be divided as many times as possible. What are the major functions? We can differentiate them to any cell type. For our interest, we're interested in cardiomyocytes, endothelial cells, and that's what we can now differentiate. So basically, by 10 cc of your blood, now we don't even need a skin biopsy. 10 cc of your blood can differentiate into different cell types. Here you could see you can get a beating cardiomyocyte as if it is beating in an animal model. You can differentiate them to endothelial cells, which has a typical cobblestone-like appearance, how we get from primary endothelial cells. And with increasing technological advances, we can get up to 95% pure, still non-mature cardiomyocytes and endothelial cells, which gives us high efficiency. We can take your 10 cc of blood, in about a couple of months you can have a look of your own cardiomyocytes which are beating under the microscope. They share the whole same genomic profile as yours, which makes it easier for us to study them because they are sharing the same genomic background with this. So once we were able to make these iPSCs from these patients, our first goal was to differentiate them to endothelial cells. I'm going to get to the cardiomyocyte aspect as well. But once we differentiate it, it's a basic protocol. You take these iPSCs, pass them through different chemical small molecules, put the endothelial medium, and in about 12 days, you max or fax sort these endothelial cells for markers such as CD31, and you have a pure population of endothelial cells derived from these iPSCs. So now I don't need to trouble the patient of taking a blood sample or a small vessel to kind of capture endothelial cells. I have an endless supply of endothelial cells from one vial of iPSCs. These endothelial cells can be passage. You could have a look now between control and disease, all have a cobblestone appearance. They're all endothelial cells. And now once I have these endothelial cells, the goal is, can we characterize these and see if they have any deficiency, knowing that they have clinical endothelial dysfunction. So first thing, we just look at endothelial markers to see, to begin with, do they have decreased markers? You could see CD31, eNOS are impaired in the disease compared to the control. What are the functional aspects? This was looking at the angiogenesis. One of the main important functions of endothelial cells is they can form tubes when placed on matrigel, kind of looking at angiogenesis. You could see all the red, the patients had impaired angiogenesis, suggesting that the endothelial function is impaired, parallel to what the clinical endothelial function is. Another function of endothelial cells is to produce nitric oxide, because that's how it causes vascular smooth muscle cell relaxation. And we can measure this in a dish by converting them into nitrate and then capturing them through fluorometric purposes. You could see they all have impaired nitric oxide production, similarly having impaired LDL uptake, which is another function of endothelial cells. So now we could see by 3 different functional aspects, all the endothelial cells from the disease patients have impaired functional properties. Now, you'll ask me the question that, is this impairment because of the mutation, or it could be because they are 60, 65 years old. Aging could be. So what we did is we genome edited this mutation. And for that, we did 2 aspects. We took a disease line and we corrected this mutation. So we called it now a wild type. And we took the control patient and inserted the same mutation. So now we can call it as a mutation line. And what this could help us is to determine whether it's the mutation which is causing the disease phenotype. And if you can look at it, that the disease line which showed impaired angiogenesis is now flipped, which is able to form tubes. That means it is the mutation which is causing impairment of the function. And the control line, which was forming tubes, is now not able to form. So it's the mutation which is causing. We went ahead and we measured nitric oxide, LDL uptake, kind of determined that it is the mutation which is responsible for this downstream endothelial dysfunction. But said that, what is the mechanism? That was my question 2. First was to characterize. So we characterized that there is a dysfunction. There is a deficiency of endothelial function. But what is the mechanism that leads to this kind of dysfunction? And for that, what we did is we took these iPSC-derived endothelial cells and we sent it for sequencing, because that could tell us at a 30,000 feet level which are the genes which are downregulated, especially in the endothelial cells. And what we did is we compared it to the control patients, and we got about 2,000 different genes which are differentially regulated. But looking closely at the endothelial markers or the genes which are downregulated, one of them which popped up was KLF2. And I'm going to try to explain why KLF2 was an important aspect for this. And these are bioinformatic tools. We try to figure out why there is deficiency in endothelial function. Now, before I tell you why we targeted KLF2 in addition to different, we first had to validate that it is KLF2 which is downregulated. So what we did is we, in addition to RNA sequencing, we did some Western blot, and you can see that the KLF2 was downregulated at the protein level as well as some chip PCR to show that it was the chromatin promoter region of the KLF2 which is non-functional because of that KLF2 gene expression is downregulated. Another aspect we wanted to see is whether its KLF2 is responsible for the endothelial function downstream of the mutation. And for that, what we did is we knocked down KLF2 in the control lines. And after knocking down KLF2 in the control lines, you could see now that they have impaired angiogenesis. So it's not only the mutation, but downstream of the mutation, it is the KLF2 gene which is responsible for the impaired endothelial dysfunction in these patients specifically. We did nitric oxide, we did LDL uptake, all suggestive that it's a step ladder pattern between lamin mutation, KLF2 downregulation, and endothelial dysfunction. Now let me tell you why we targeted KLF2. This goes by flow-mediated condition. So as I said, the endothelial cells in your vessels are always in a dynamic state because blood is always flowing through this. This is your normal flow hitting the endothelial cells. What it does is that it stimulates a bunch of mechanoreceptors. So your endothelial cell is the best mechanotransducer. If you decide to run from here to the other room, your endothelial cells will detect the increase in your flow, the blood, which is by the heart pumping, and it can send information downstream to the smooth muscle cells, which can help relax so that you have more blood flow passing through your skeletal muscle in case if you decide to run. So what it does, it hits the mechanosensors, and it sends information in the nucleus, which is upregulation of KLF2, which then upregulates eNOS, which is NOS3, to produce nitric oxide, which is a gas, gets into the smooth muscle cells and causes vasorelaxation. What it was missing is that it has to go through lamin. So lamins is at the cross junction. It's at the crucial point of upregulating KLF2. And that is the reason we wanted to go after KLF2, because our central hypothesis it was that KLF2 might be intrinsically downregulated in this laminopathy patients. But how do I prove it? Because all my conditions so far were in static conditions. We were not doing flow-mediated conditions. We had cells which were growing in the dish, but just sitting there in a static conditions. So what we did is we used the IBD engineering system where they have this beautiful system where you can subject your cells, and for us it was endothelial cells, through a flow-mediated system where you could run medium blood through these cells for a week, which is kind of mimicking how our vasculature works, where blood is flowing on a continuous basis. And this would prove us whether KLF2 is important, kind of a second validation. So what we did is we took iPSC ECs from both the patients, control and disease, subjected them through a flow system for 24 hours, and we kind of mimicked what our physiological flow would be, about 10 to 15, 5 to 10 dynes per centimeter square. We lowered it down because these are iPSC ECs that are more on the immature side. So ramping up the speed usually caused the cells to slough off from the dish. Something that we are working on to make it more physiologically relevant. So what happened with the flow-mediated system? So if you look at it, this is your control cells. And you could see how the endothelial cells in the slide is in a line with the flow. This is how the medium is flowing. However, the disease line, you could see it's all over the place because it's dysfunctional. There are downregulation of KLF2, which we saw through the RNA sequencing study. However, when you genome edit, you could see it is reversion. So it is the mutation which is affecting KLF2, but once you reverse the mutation, KLF2 is restored and alignment comes through. So we run it through, and then we did angiogenesis and nitric oxide production. And if you could look that not only in the static condition, but in the flow condition, The control had more angiogenesis compared to the disease, same thing with the genome edited. So now we can prove it is the KLF2 which is responsible for the impaired endothelial function in it. Now, as I said, now I have a target. Yeah, go ahead.
Was there anything surprising about pulsatile versus continuous flow?
What we found is that there was more, so there was. So KLF2 in a static condition is not upregulated. It is only through the flow-mediated system we see a KLF2. And that's why here it's not on this slide, we looked at KLF2 in the flow-mediated system. In the static conditions, KLF2 is always in a downregulated system. So it was not a fair system to compare and measure the sequencing or the downstream studies. So that's why we switched to a more physiological system. So definitely there was a difference. The endothelial dysfunction was more or less consistent. Even though it is not through KLF2, again, as I said, there were about 180 genes which were downregulated in the endothelial cells in the sequencing. So I'm thinking it's a combination of different genes which are downregulated, which are affecting the endothelial function, not just KLF2. Yeah, go ahead.
So in your schema, you show that endothelin and nitric oxide are two downstream signaling pathways from the KLF2.
Right.
And there's a bunch of really good pharmacological inhibitors of both. So did you look at those two pathways?
So we did. So first we knocked down KLF2 by using shRNA, but then we did ENOS, we were just talking about it, by using ENOS inhibitors to see whether it is ENOS inhibiting in the control line, whether it will impair endothelial or vascular smooth muscle cell relax. So we did that. But our goal was to see if we can enhance KLF2 in these patients so that I can bypass the lamin mutation and see I can restore the endothelial function, but just improving KLF2. Because then there is a way I can improve the function of the endothelium in response to having the mutation. So we did check that. We wanted to check every aspect of this whole pathway. We knocked down KLF2 by using siRNA. Then we knocked ENOS by using inhibitors. All of them had a sequence effect in this thing, in the genome-edited line as well as the control lines.
And endothelin?
We didn't go, So endothelin has a different aspect of the vasoconstrictor. We wanted to look more on the vaso relaxation aspect. We didn't check for the ETV1. We didn't, yeah. So as I said, we had the target. So, apart from the different genes which were downregulated, KLF2 was the one which we were really interested in, especially knowing that we kind of validate that in our flow-mediated system where KLF2 is very important. We wanted to screen for molecules which can enhance KLF2 in the disease patients. Lucky for us, we didn't have to screen using a high throughput method. What we did is the first thing we did is we use ChemBank or MedBank. These are banks where people have tested different compounds for different targets and upload the data. So ChemBank was a very good resource. It's part of the Broad Institute at Harvard. All you need to do is put the target in it. It will spit out a number of different compounds which can regulate. For us, it was as simple as adding KLF2 regulators, and it spit out a bunch of molecules. All of these were regulators of KLF2, and we had to do is do a simple assay of looking which of these can improve KLF2 expression, both in the disease line and the genome-edited lines. And if did improve KLF2, that could be a potential target of improving endothelial function. So what we did is, and not to show the entire set of data, we went one by one for each of these drugs and looked for KLF2 expression for both in the disease line as well as the disease genome-edited line. And we found 3 of these drugs right at the top to improve KLF2 in a dose-response manner. And this is something which Mukesh Jain at Case Western has already proved, that lovastatin, mevastatin, and simvastatin can improve KLF2 expression in endothelial cells. So it was something that we wanted to improve KLF2 expression. We proved it by looking at KLF2 in our genome-edited as well as the disease lines. So the question is that now we know that lovastatin or mevastatin can improve, can I recapitulate in a dish? And that's exactly what we did, is we took all the 4 different lines, we put them in our flow-mediated system, and this time we threw in lovastatin or DMSO to see if we can reverse the phenotype. So now if you look at, this was the original figure which you saw, that this is misalignment, but the moment I threw patient 2 with lovastatin, they had more of an alignment, because now I am able to have KLF2 being restored, which can now allow the alignment. But importantly, what we could do now is we can improve angiogenesis. So this is the disease line, and the moment we put lovastatin, you can see there is improved angiogenesis, and there is improved nitric oxide production. Now, this is one way we can bypass the mutation and go downstream to improve KLF2 expression. So we knew that we have a target, and we have an FDA-approved drug which can improve, at least in the dish, the phenotype of the endothelial cells. Now, as I said, eventual goal is to see if we can then model the disease. We have screened for drugs. Is there any way we can get it back into the patient, especially knowing that we got lucky that it was an FDA-approved drug? Now, remember, before I started the study, they had clinical endothelial dysfunction. We measured with EndoPAD. So we knew that they have impaired endothelial dysfunction. Now, is there any way I can give these patients lovastatin, which is already an FDA-approved drug, and then try to measure their endothelial function? So we measured just before giving lovastatin, we measured the EndoPAD again, and we found consistent results. They had endothelial dysfunction. And then 6 months later, we followed up with the patients, and now we found not a complete recovery. It wasn't 1.67 and above, but there was improvement in the endothelial function, and we did this repeatedly. One of the patients, this one, underwent heart transplant because his case, so he dropped out of the study at 8 months. But as recently as last week, patient 3, who was on lovastatin for about 1.5 years, came back, and we measured him. We have seen a value of increase from 1.08 to 1.31. So this is in a minuscule, just 2 patients. But it does show that there is an improvement in the patient itself. Now, we saw rescue, and you will tell me that what happens with these patients then in primary tissue. So we got lucky with one aspect of this. So patient 2, before we started with lovastatin, had gone some valvular replacement. And the surgeon was able to give us a small nick of an artery where we could measure the endothelial cells. We isolated the endothelial cells, and you could see that they were impaired angiogenesis. 6 months later, he underwent heart transplant, and we got the entire heart. And now the first thing we did is we isolated endothelial cells 6 months after lovastatin, and now you could see there was much improvement in endothelial function before and after. So for another patient, the patient 3, we were able to collect blood ECs before and after 6 months of lovastatin, and you could see there is much improvement in angiogenesis before and after the drug treatment. We didn't have enough cells to measure nitric oxide production, because these primary tissue, it's very difficult to isolate a ton of endothelial cells. Usually they are valid only for 1 or 2 passages if we get lucky. iPSC-ECs, we could go for 6 or 7 passages. But another study we want is, so I said endothelial cells secrete nitric oxide, which has an influence on the smooth muscle cells. What happens, is there any way we can measure the nitric oxide production on this thing? So as I said, we had one of the patients who underwent heart transplants, and when we get the heart, we isolated the LAD from this human heart, and we put it on this device which is called a myograph. So the myograph, you can measure the response of the endothelial cells on the vascular smooth muscle cells, and in a way you can quantitate the amount of nitric oxide being produced. And what we did is we took this laminopathy patient and we threw it on the myograph in response to acetylcholine. So acetylcholine is a drug which will cause nitric oxide production, and it can measure the capacity of the endothelial cells to produce nitric oxide by measuring the vascular smooth muscle cell relaxation, which you can then calculate as % relaxation. It's the most accurate way of measuring the nitric oxide response on vascular smooth muscle cells. So you could see the red line has impaired vascular relaxation. But 6 months later, when this patient came back, you could see with the pink line had a little bit more improved response, means the endothelial cells had a little bit more better response to acetylcholine producing nitric oxide. Here, for the control patient, we had a patient who was had a car accident. We were able to get the heart recovered, and we threw in, and you could see that it had the best. It was a healthy control. It had the best response to acetylcholine. So the bottom line to say right here is that lovastatin given to these patients not only improved endothelial cells, but it also improved vascular smooth muscle cell response. Now, my slide here said, and I'm trying to wrap it up, my slide initially said all roads lead to Rome. And how do I tie this to the heart? And that's the question which we wanted to solve, because if I can improve the endothelial health, how am I going to improve the cardiac health? Because that's what these patients usually have. Of cardiomyocyte deficiency, they're not able to have increased contractility to have the capacity to pump blood out of the heart. So what we did some co-culture studies where we took the control and the disease, and then we co-cultured the cardiomyocytes and the endothelial cells with or without statins, and see whether improving the endothelial health with statin can improve the cardiomyocyte contractility. Is there any way I can improve the cardiac contractility, which is a major impairment in DCM patients? And long story short, you could see that only when the iPSC cardiomyocytes and the endothelial cells were co-cultured and treated with lovastatin, you see this red one had improvement in its contraction velocity, had much better amplitude in calcium, which shows that it has a much better contractility in the heart, and a decrease in the diastolic calcium. So in a way, if this lovastatin can be given long term, is there any way I can improve the contractility of these patients, which have a much better outcome for its dilated cardiomyopathy? Now, said that, can I reverse the mutation and the dilated cardiomyopathy? Probably not, because the disease is already set in. Usually the outcome is a heart transplant. That's what we need to do for these. But think about the younger individuals who are 15 or 16-year-old who has this mutation. Their onset of disease is not going to be till the age of 50. Can they start on a statin much earlier to have a better outcome, or maybe delay the onset of the disease? So that's something which we were going after, that maybe the younger individual who has the mutation but not yet the disease can start on lovastatin to have a better outcome at the age of 50. But then, we want to, kind of delve a little bit deeper into it. I mean, and this slide shows it well, that all the study which I showed was in a 2D. Can we convert this from 2D and 3D and beyond, which is kind of sort of like a 4-dimensional? Because that would tell me more accurately what is this co-culture between cardiomyocytes and endothelial cells can be achieved. And for that, we have two different ways we deal with. One, we have cardiomyocytes and endothelial cells. We co-culture them together, and you could really see how well this purity is. This has iPSC-derived cardiomyocytes with endothelial cells. The moment we put endothelial cells with the cardiomyocytes, they beat more vigorously. There's a little bit more maturement of the cardiomyocytes. And once we have these co-cultures, we can make two different 3-dimensional structures. One is called as the human cardiac organoid, and something called as the human engineered heart tissue. They have all the 3 cell types which are present in the heart. The fibroblast, the endothelial cells, and the cardiomyocytes all derive from iPSCs. And this is what my research is now pivoting towards, because this would give us the best indication of how a heart tissue works in. They are from the laminopathy patients, and it gives us more information of what we can use for drug discovery, to understand the disease model much better than in a 2-dimensional structure. What we have also done is, once we have generated from, and this could be not just for laminopathy patients, for diabetic cardiomyopathy patients, where we can subject them to hypoglycemic conditions, or chemotherapy, they eventually develop cardiomyopathy, we can expose them to doxorubicin. But eventually the goal is to understand and study them. And this is engineered heart tissue. I just wanted to show how brilliant it looks when you stain them for fluorophore. This is from a disease line, and you can see how rigorously it is beating. It's an arrhythmic potential with different calcium imaging on different aspects. But eventually, if I want to really do something on a clinical trial in a dish, I need to have it on a more four-dimensional where different organs can be combined together. And what we're doing is, there's a company in Germany which makes these chips. It's a three-dimensional chip where your idea is you could put your engineered heart tissue in one chamber, the endothelium in one chamber, or other cell types, and then pass flow from these different systems with different drugs. So now you have a sort of not only a three-dimensional, but a four-dimensional system. Rather than doing these drug testing on individuals, you could do on the dish, come up with a much more informed which drug is going to be before using it on a clinical system. So we are implementing these different aspects. I added these slides because I'm coming to Virginia Tech. I wanted to make sure that I show some of my engineering aspects as well. So we could use this as a clinical trial in a dish. This is just showing some images. With that, what I showed you today, and we're doing this not just for an inherited heart disease, we're also doing this for diabetic cardiomyopathy, for chemotherapy-induced cardiomyopathy, iron overload, where it has a big impact on cardiomyopathy. We can recruit these individuals. We can generate these iPSCs, differentiate them to different cell types. And this is not restricted only to cardiac cells. Neuronal cells is a big thing where people are studying Alzheimer's, studying different kind of neuronal disease. You can model the disease, genome edit the disease, figure out the mechanism, look for a target, screen for a drug, and then if all of this is successful in the dish, you can then run a clinical trial knowing which drugs show improvement in the dish. With that, I think I'll thank all of you guys for listening. That's my director, Dr. Wu. He's the director of the Cardiovascular Institute, my funding sources, which has helped me with these last 8 years of my postdoc instructor career. And this was one of the engineered heart tissue. When we took it off the post, it kind of curled into what I look like a heart. But it might not, but it was beating. And I was like, this could be good for a job talk, where I can show this as a heart. With that, I'll take any questions if you have. Yes. Yes.
Very nice, very interesting stuff, really nice work. So I'm just trying to connect the endothelium to the cardiomyopathy, the systolic dysfunction,
Right.
dilated cardiomyopathy. And clinically, we see lots of patients who have terrible endothelial dysfunction, renal failure patients.
Right.
When you do these vasodilator, the Flow-mediated vasodilatation studies, they have terrible endothelial function. And yet many of them have perfectly
True.
normal contractile function. So I'm still not sure I see the connection.
Sure.
Moreover, one of the mediators that you look at, nitric oxide, there are a number of reports, unless I've missed some recent literature, that nitric oxide is actually a negative inotropic factor in many studies for contractile function. So I'm just trying to, how it is that you connect endothelial dysfunction more closely to the LV dysfunction, or certainly you proved that there's endothelial dysfunction without a doubt. I'm a believer.
Right.
In these LMN patients, but I'm not seeing the direct connection to the cardiomyopathy.
So let me answer by this specific family. The entire family had diastolic dysfunction. So that's what we picked on, is that when we had an entire paper on the cardiomyopathy aspect as well, which got published earlier in the year. But when we were looking at these patients, the clinical aspect, we found a vascular phenotype, and we found they had a diastolic dysfunction when we measured through MRI. And that kind of made us think that maybe these patients have an underlying vascular dysfunction which is contributing to the heart. And that led us to think that maybe we should investigate the endothelial cells. Now, why did we do? Because lamin is present in every cell type. And lamin has been known to cause endothelial dysfunction in n number of diseases. For example, progeria. And this is why we focused on the laminopathy patients, where we can connect the endothelium to the cardiomyocyte. Because the lamin mutation is in both the cardiomyocytes and the endothelial cells. Now, connecting them together was our afterthought, where we thought that is there any way, if we improve the endothelial function in this laminopathy patient, will that improve? Now, coming to the question is, does nitric oxide? So there are two schools of thought in this. One is that if nitric oxide in the vicinity of the cardiomyocyte, it can improve the contractility of the heart itself. Because what it does, it causes a lot of S-nitrosylation of different cardiac proteins, which will cause improved calcium handling within the sarcoplasmic reticulum and the cytoplasm by the nitric oxide which is secreted within the vicinity. And that's what made us to connect those two things together.
But isn't there also pretty robust literature that nitric oxide has
True.
a negative ion?
Well, depend on the concentration of the nitric oxide. So let's say if there is, nitric oxide produced by the endothelial cells in a physiological condition and a pathophysiological. So if you have too much nitric oxide being produced, it can combine to form reactive oxygen species, reactive nitrogen species, which has a negative inotropic system. So that's where the balance comes in. So endothelial cells, when they are physiologically relevant, they secrete nitric oxide, they're secreting all these good factors. But when they are tilted towards a pathophysiological, ETV1 is one which is produced, TGF-beta, they are secreted by the endothelial cells, which has a negative impact on the heart itself.
If I could ask just one more quick question. The statin effect, I presume, is not a cholesterol-lowering effect. Have you looked at the small g proteins, the farnesylation, geranyl?
That is a good question. So is this effect because of lowering of, LDL and lowering of all of those, and not just because it has an effect on the KLF2? So we did check all of those factors. Indeed, it lowered the LDL and all of those factors for these individuals, but what it also does is that KLF2 was upregulated. Now, one of these patients was on atorvastatin, and we switched him from atorvastatin to lovastatin. When he was on atorvastatin, he had clinical endothelial function. But when we switched him to lovastatin, he showed improvement, and this is because it has a double effect. Atorvastatin cannot improve KLF2 expression. But when we switched it to lovastatin, it has a double effect. Not only it can lower your cholesterol effects, but also improve KLF2 expression. So we did check that. This effect is specifically because of LDL improvement, or is it because of KLF2? And I'll answer another way, because we submitted this paper to Science Translational Medicine, and they came back with similar questions. I mean, we did the revisions now, we're about to submit. Their question was, is it because these individuals have massive atherosclerosis, that's why you see endothelial dysfunction? Or is it because of the lamin mutation? In a way, then statins had an effect. So we recruited 2 of the young individuals who have no atherosclerosis. They have normal LDL level, normal cholesterol level, and they also showed endothelial dysfunction. We're trying to recruit them so we can give them lovastatin and see whether now they can improve endothelial function, even though they have normal LDL levels. That would prove that it is not because of that, it is because of improving KLF2 in the endothelial cells. Yes?
So, thank you. In your co-cultures, how do you assess, this is kind of a follow-up from our morning conversation, how do you assess cardiomyocyte maturity? And then as a follow-up to that, do you ever see like hypertrophy in the cardiomyocytes? Do they go from mature to pathologically?
So iPSC-derived cardiovascular cells, they're all immature. We had a great conversation of how we can improve. This is throughout the field, they're all immature. Now, adding endothelial cells in general to cardiomyocyte, iPSC-derived cardiomyocyte, improves its maturity, but it's a very incremental level.
How do you assess it? Is it by the same way you did in the beginning of just looking for the genes that are...
But that's all we can do right now is we haven't, and that's where we have to do MEA or some kind of electro to see what the maturity level is. Adding 2 different cell types does improve the maturity of these things, but not much. And what we're doing is looking at the RNA or the different gene expression pattern to see whether the cardiac function is improved. And then what we could do is measure contractility or you could do calcium handling. What is missing out of the study is electrophysiology. And I'm so glad the reviewers didn't ask for it, because that is quite, I mean, it's a tedious process. We had 16 different groups with or without statins to see whether it is only through statins effect which is happening. And now, statins don't work on cardiomyocyte directly. They don't have the, it has to be through the endothelial cells. I think once we get into the engineered heart tissue, I think we have a better approach of understanding these co-cultures, because now they are meshed together rather than adding two different cell types together. Because then they have a more of a physiological heart tissue. I mean, they are organoid, or they are these EHMs.
Yeah, right. I'm curious, when you do the co-cultured cells, if you're able to achieve that 3-to-1 ratio that you talk about.
So we played around a lot for that. That took us a long time. We started with 7:1. So remember, the cardiomyocytes will not proliferate, but the endothelial cells will proliferate. So we started with a 7:1, 5:1, and eventually we reached that it was 1 endothelial cells to 3 cardiomyocytes. Because in 1 week of treatment with statin, that 1 endothelial cell, more or less, I mean, this is just hypothetical, because we were fact sorting and counting how many cells we get. Eventually, 1 week of treatment, that 1 endothelial cells would become 15, so that every 5 cardiomyocytes, above 15 endothelial. So we played around a lot with them, because we don't want to inundate with too much endothelial cells, because then the effect of cardiomyocyte cannot be measured. Because then eventually these co-cultures were measured under image-based contractility. And if there's too many endothelial cells, you won't see these beating cardiomyocytes.
Do they need to be in direct contact?
In a way, yes, that they need to, because the nitric oxide needs to be produced, has to be close to a cardiomyocyte. Now, we didn't go into how close they were, but I think, again, the answer would be, if we can do 3D constructs with them, they are enmeshed in this thing. They are formed when you make them together on an agarose construct. They are together. But here we were just putting them together on Matrigel, swirling and mixing it, and hoping they're close to each other.
Yeah, I mean, maybe that German machine will.
So that would be where we have the organoid, and we have the endothelial cells in the lining of the tube, and then we put the flow system, that how the endothelium with the organoid, the cardiac organoid, works. But that's the next step.
John. So I have a quick question. Actually, would you mind going back to your RNA-seq data?
Yeah.
The curiosity on some of that data, because I was just glancing through the top candidates. There's not too much trouble.
This is preliminary one, which I put. We have done, yeah.
Yeah, so while you're searching for, maybe you can answer with that.
Sure.
Static conditions.
These were static conditions.
So the curiosity there is, you said KLF2 on cells not experiencing flow is already little to nothing?
Right, so that's a good question, because what we did is we repeated RNA sequencing on the flow-mediated system. But if you look at this, the control, so all of us have some basal level of flow. Even if in a dish, you're going to change medium every day, which is having some kind of an effect. Everything was compared to the control iPSC-ECs, which were in a similar condition. And that kind of gave us an idea that KLF2 was downregulated. Of course, it was not in a flow-mediated condition, so we repeated the entire RNA sequencing through that chamber for 24 hours, picked up. What I didn't show is that we did some attack-seq as well, because we wanted to compare the chromatin of KLF2, the promoter, or the mutation having an effect on the KLF2 promoter, which has an effect on the gene expression. And we lined the attack-seq, which is looking at the chromatin, and the RNA sequencing after flow together to see if KLF2, it's the integration of the attack and the RNA to spit out KLF2. I didn't show that data because lack of time, and I wanted to dwell. But remember, lamin is so important for epigenetic changes. It is the information which is being passed from the nucleus into the cytoplasm to the nucleus.
The other thing I want to ask just quickly is that you said PKM1 and B kept here and there. To me, it almost speaks of another function of endothelial cells, and that's barrier function.
That's true.
Right? It doesn't seem,
Well, we had about a bunch. This is just a table I put up saying that the top 3 were PCAM CD31, CD144. Some were flow-mediated, and towards the bottom, if you see, they're more like integrin. We have about 180 endothelial-specific downregulated genes in the disease line. Overall, from the 10,000 genes we got from RNA, about 2,000 something were downregulated. I mean, lamin is such an important gene, it has an effect on a bunch of other factors as well. But I'm just putting as a very simplified table to kind of steer you towards that KLF2 was something which we popped out. And honestly, KLF2 is because of two reasons. One, we measured their clinical endothelial function first. And the first thing, which is flow, is very important for KLF2 to go up. And secondly, the literature out there that eNOS production, the eNOS upregulation and nitric oxide has to be dependent on KLF2. So this was like an easy slam dunk for us to kind of chase after. And then luckily we found there was an FDA-approved drug. We were measuring clinical endothelial, which is flow-mediated, kind of tied it together.
I'm just asking one
Yeah.
little follow-up question. It was related to David Saint's question earlier. So with these concentrations of NO, I take your point. If you change the environment in terms of superoxide production, and you're limiting peroxynitrite production in that area, you don't get the post-translational modification downstream. Does that affect the overall response?
It could. We didn't. So another aspect. I'll try to answer it this way. These patients have a lot of inflammatory markers. We checked that on the EC level as well. So there could be a possibility that because of the mutation, there is some dominant negative effect where they have all these peroxynitrite in the flow. Their flow is maybe not that stable. It has some turbulence, which is causing some ROS production. We didn't check the effects of that on the co-culture. We could do that. We could do it on a three-dimensional level where instead of having a more linear flow, we can have a more turbulent flow. That can produce ROS production, which can have an effect on the cardiomyocytes. We didn't check that.
Okay, thank you very much. We appreciate it.
Thank you.